Types of genetic linkage analysis. There are two main types of the genetic linkage analysis. They are called as parametric linkage analysis and non-parametric linkage analysis. There is another classification that how to classify the genetic linkage analysis. That classification includes dichotomous linkage analysis, continuous phenotype, heuristic linkage analysis, two-point linkage analysis, and multi-point linkage analysis. In case of parametric linkage analysis, usually this analysis is based on the traditional approach to search the probability that a gene for a disease is linked with the genetic marker. So in this case, we usually try to identify that the disease gene is linked with the genetic marker, and the phenotype and the marker, they co-segregate with each other. Usually in case of parametric linkage analysis, Lord score is calculated. Lord score tells that whether the phenotype and the markers, they are tightly linked. If there is a higher Lord score, then there is a high probability that there is a gene for a particular phenotype. While in case of non-parametric linkage analysis, that studies the probability of an allele being identical by descent or by the ancestors. So this is the second type of the analysis, which is called as non-parametric linkage analysis. In this case, usually there is an association of the marker with the phenotype. In case of parametric linkage analysis, Lord score is calculated. Positive Lord score tells or it favors that the presence of the linkage, while the negative Lord score tells that there is likely to be less linkage. So based on the positive Lord score and the negative Lord score, linkage analysis is confirmed. Computerized Lord scores are calculated, or that is a simple way to analyze the complex family pedigrees in order to determine whether there is a linkage or there is no linkage. In case of non-parametric linkage analysis, if a trait locus truly regulates the expression of a phenotype, then the relatives of the individual would have similar phenotype, should have similar genotypes. So in this case, in case of non-parametric linkage analysis, usually if there is genotype of the individuals, then based on the similar genotypes, there should be similar phenotypes. So such a association is identified in this case of non-parametric linkage analysis. Correlation between the phenotype similarity and the marker genotype similarity is identified in case of non-parametric linkage analysis. No need to specify mode of inheritance or the allele frequencies. So it means in case of non-parametric linkage analysis, there is no need to check or to confirm the mode of inheritance or what are the allele frequencies in case of non-parametric linkage analysis. In this diagram, we try to understand that what is the difference between the parametric and non-parametric linkage analysis. Here is the chromosome, here is the gene, and here is a marker. So if the phenotype has pure genetic factors, on the other hand, the phenotype has pure environmental factors. In case of genetic factors, if there are strong genetic factors, then usually a recombination frequency is identified between the marker and the phenotypes. And usually mode of inheritance is also checked before performing the linkage analysis. While on the other hand, if the phenotype is due to the genetic factors as well as environmental factors, then usually a correlation or association is identified between the marker and the phenotype. So this is the difference between the parametric and non-parametric linkage analysis. So if we conclude that what is the genetic linkage analysis? Genetic linkage analysis is of two types. The first type is called as parametric linkage analysis and the second type is called as non-parametric linkage analysis.